This is you. Not the frog. You're something much smaller.
Yeah, that tiny zoospore is you. Bactrococytrium dendrobatitis. You're in the first stage of your diphasic lifestyle. For the first portion of your life, you're a waterborne motile unicellular organism about 3 to 5 micrometers in diameter with a long posteriorly directed flagella that can be up to 20 micrometers long. You lack a cell wall at this life stage, but don't worry, that'll come later. The fungi species found within the class Chytridiomycetes, to which you are a member, are found in soils, freshwater, and marine environments. Like your closest cousin, Bactrococytrium salamandrovorans, you're found in primarily aquatic environments, but you can survive in moist soil for a small period of time. As a zoospore floating around in the water, your primary goal at this stage in your life is to find a suitable host. Members of the Chytridiomycota phylum are ubiquitous aerobic saprotrophs, chemoheterotrophs that get nutrients from decaying organic material. As B. dendrobatitis, you take being a saprotroph to the next level, as a parasite. While swimming around in your aquatic environment, receptors on your cell membrane pick up the presence of nutrients, such as the amino acids cysteine and glycine, the protein keratin, or sugars such as glucose or lactose. You'll follow this nutritional trail through the water, positively chemotaxiing toward the source. Target acquired. Yes, this looks like the perfect place to set up shop. Once you touch the skin of an amphibian, you'll go through a process known as encystation. During this time, you'll retract your flagella, losing the ability to move around the environment, and build a chitin-based cell wall to encapsulate yourself. It's thought that mucus components on the skin of amphibians called mucins trigger the metabolically expensive encystation process. Vesicles within your cell hosting the chitin synthesis are blocked from the cell membrane by actin molecules. When mucin reaches receptors on your cell membrane, it causes a spike in the intracellular calcium that is theorized to remove the actin barrier, allowing the chitin-forming enzyme to fuse with the cellular membrane. This, in turn, leads to the formation of your brand new cell wall. This calcium signaling process leading to encystation is unique to yourself and your cousin, B. salamandrivorans. Other members of your class have different triggers for encystation. During this time, you'll also have to fight off the security system of your new host. Dermal glands on the skin of frogs secrete antimicrobial peptides, also known as AMPs, that are hypothesized to disrupt your cellular membrane before the cell wall can form. Rude. You'll also encounter antifungal metabolites from the bacteria that already call this area home. Sometimes the two will even team up and pick an uneven fight. You can't always win, but it looks like you came up on top today. Nice. Congrats! You've been promoted! You're now considered an immature zoosporangium, sometimes referred to as a trimling. Now that you've found a host, it's time to get nice and comfy. As you invade the epidermis of your new amphibian neighborhood, you settle in on the deeper stratum basale. This is the deepest layer of the skin, and all the cells around you are still living, so you'll hunker down here and focus on growing into a more mature sporangium. You'll use a germ tube to spread your genetic information into your new host cell before the deep end of the tube begins to swell, forming the thallus. You'll increase 2 to 3 times in size as your cytoplasm becomes more and more complex. You originally had one nucleus as a zoospore, but through mitotic divisions you become multinucleated in preparation to produce new zoospores. Not only are you growing, but you're moving closer to the surface. As the skin of animals grows, old layers are shed off the top and replaced by lower layers. The lower layers of the skin go through a process called keratinization, which leads to the death of the skin cell as they move up toward the surface. Scientists think that the timing of your maturation is related to the rate at which deeper skin cells undergo keratinization and lose all organelles, giving you more room to spread out and grow within the skeleton of the now superficial dead skin cell. During this time, you also develop at least one discharge papillae leading to a discharge tube. Depending on how deep you are in the stratum corneum, that outermost skin layer, zoospores can be released into the water or into the spaces between epidermal cells. The new zoospores are then free to find a new host or reattach to your own amphibian neighborhood. So now you're an empty nester, and there's not really a whole lot left for you. You've served your purpose, and you remain empty. You're now clear in color, and your cell walls may remain or they might collapse. Other bacteria might find your discharge tube, follow it down, and colonize within your now empty thallus. Occasionally, a zoospore might decide to live with their parents for a bit longer and go through the development process within your empty sporangium. You're Mr. Worldwide! While you were first identified in Australia in the 1990s, it's thought that the most prevalent pathological strain of B. dendrobatitis, named BDGPL for global panzootic lineage, has its origins in the Korean peninsula, before you spread everywhere due to human activities and the increased global amphibian trade. There are 4 other lineages, BD Asia 1, BD Asia 2, BD Asia 3, and BD Cape, that can be found in various parts of the world, but they're not everywhere like yourself. There's a lot of information still not known about how exactly you get your nutrition, why you grow with inconsistent growth rates, or if you can even exist as a non-pathogen like many of your cousins. So why do people care so much about little ol' you? Ah, well, that makes sense. Listen, you might be responsible for up to 90 known extinction events, and just maybe driving countless other species toward the brink. But it's not your fault that you're found in over 700 different species of amphibians. You can't blame a fungi for just living their life, disrupting the osmoregulatory functions that occur in amphibian skin, leading to electrolyte imbalances, possible secondary infections from other pathogens, or cardiac arrest, can you? Oh, you can? Huh.
Hey everyone, Gabby here, and today I'll be discussing Candida albicans, a fascinating microbe that is both a normal part of the human microbiota and a potential pathogen. I'll explore how it switches between harmless and harmful forms, its role in human health, and why it's particularly interesting to me as I pursue a career in ultrasound technology. Candida albicans is a common fungus found in the mouth, gut, and vagina as a part of the normal microbiota. It usually coexists peacefully with other microorganisms. However, when the body's conditions change, like a weakened immune system, it can become pathogenic and cause infections ranging from oral thrush to invasive candidiasis. Candida albicans is dysmorphic, meaning it can switch between a harmless yeast form and an invasive hyphal form. In its yeast form, it's non-pathogenic, but when the environment changes, it becomes more dangerous by switching to hyphae, which allow it to invade tissues and cause infection. In yeast form, Candida albicans is small, round, and exists harmlessly in the body. But when the conditions are right, it changes to a filamentous form called hyphae, which are long, unbranched filaments that invades tissue. These hyphae also form biofilms, making it resistant to immune defenses and antifungal treatments. Candida albicans is diploid, which helps it adapt to various environments. Metabolically, it switches between fermentation in its yeast form and oxidative metabolism in its hyphal form, allowing it to survive in areas with different oxygen levels, like the gut, mouth, and vagina. Candida albicans interacts with other microbes in the human microbiome. Normally, it exists with bacteria and fungi maintaining balance. However, disruptions like antibiotic use can promote C. albicans overgrowth. Some bacteria inhibit its growth, while in other cases, it can support the growth of certain bacteria. As an opportunistic pathogen, C. albicans is usually harmless, but it can become harmful when the immune system is weakened. It switches to its hyphal form, allowing it to invade tissues and form those biofilms. Invasive candidiasis affects over 250,000 people globally each year, leading to significant health complications. Treatment is challenging due to C. albicans' ability to form biofilms and resist antifungal treatments. Candida albicans plays a role in human health and helps maintain microbiota balance. But when the balance is disrupted, it becomes pathogenic. This ability to shift between harmless and harmful forms shows how important microbial balance is for health. All right, this is my artistic representation of Candida albicans. For this project, I used acrylic painting to visually represent C. albicans' adaptability. I showed the painting in both yeast and hyphal form, along with chlamydospores, which are those little balls on top of the thread-like structures. I wanted to show how it could survive in harsh conditions, and so that's why I did that crazy little background to try to make it look like different temperatures. But yeah, in conclusion, Candida albicans is a highly adaptable microbe with a significant impact on health, and understanding it is crucial for improving infection treatments. Thank you.
Eukaryotes unilaterus is a eukaryotic fungus that is better known as the zombie ant fungus due to it taking over ants' behavior to self-propagate. O. unilateralis is part of the Eukaryotes genus that houses over 200 species, with more being discovered around the world in tropical environments. It is an obligate parasite to carpenter ants, specifically Camponotus formicine, meaning that it is host-specific and does not infect other insects. There are other species in the genus that infect other insects, such as different species of ants, or even other bugs like moths, spiders, or grasshoppers. The order Eukaryotes infect arthropods by penetrating the host integumentary system using spores to attach to the epicuticle or the exoskeleton, germinate on it, and then are able to grow through the procuticle, the chitinized part of the exoskeleton, which presents a significant physical and chemical barrier to infection. It does this by using pressure from hyphal growth, which is filamentous thread-like structures called hyphae. It also uses hydrolytic enzymes like proteinases and chitinases to degrade the insect's cuticle that increase the virulence of the fungus. The fascinating life cycle of O. unilateralis starts as a sticky spore on the forest floor, just waiting to attach to a passing forager ant. Once attached to the exoskeleton of the ant, the fungus grows as blastospores, which are yeast-like cells that release toxins, destroy host tissues, and will eventually kill the ant. While the fungus grows inside, it interacts with the ant's microbiome, specifically the mycobiome, which is the collection of fungi in the ant's gut releasing enterotoxins. The ant starts to develop erratic behavior and movement. It wanders off its typical path, climbing to a spot where the conditions are optimal for fungal growth, somewhere warm, humid, and with just the right amount of sunlight. The infecting fungus gains resources from living hosts from the degrading cuticle in a biotrophic phase and using secondary metabolism, meaning that it produces several chemicals that are not essential for growth, but help the fungus manipulate its host and protect against competing microbes. There are also bacteria, such as Bacillus cereus, that have a symbiotic relationship with O. unilateralis. They are resistant to the antimicrobial properties of the fungus, help break down the tissues of the ant, and take up resources so other bacteria cannot coexist. Instead of taking over the ant's brain, as the name zombie fungus would imply, it actually attacks the muscles of the ant, especially the mandibles. This helps in the involuntary act of biting behavior where the ant clamps on to the underside of a leaf and is rendered immobile until the ant dies. After the ant dies, the fungus devours the ant from the inside out and will produce a stroma protruding from the back of the ant's head to release spores into what is known as the killing zone underneath to infect more ants. The stroma is a long brownish stalk that grows spherical fruiting bodies called ascoma. Ascoma contain the asci, which are sacs that are cylindrical in shape, hyaline, meaning clear in color, and contain 8 spores each. Spores of O. unilateralis have a special adhesion structure called abrasorium that help attach the spore to the body of the passing ant. Here you can see the ascoma on the stroma with the asci inside waiting to burst out and disperse spores down below to infect more victims. Since O. unilateralis is a specialized pathological parasite that infects and manipulates and kills the carpenter ants, it is actually considered a keystone species that helps keep the ant population under control, meaning it has an important role in balancing the delicate and highly diverse tropical ecosystem. But even this zombie fungus can't go unchecked. It is known to have hyperparasites, which are parasites to parasites. One of these species is Trobiolomyces zombiae. The hyperparasites cover the original fungus and its stalk, which effectively prevents O. unilateralis from injecting its spores. They are also obligate parasites, as they are not known to grow on anything else except O. unilateralis, which depend on the zombie fungus, which depend on the carpenter ants. In this image, you can see the hyperparasite fungus growing on the ascoma of O. unilateralis. O. unilateralis is effective in keeping ant colony numbers under control, so much so that there are what we call graveyards under killing zones with densities that may exceed 25 ants per meter squared. Here's the collage I made of O. unilateralis, where you can see a carpenter ant fixed to the underside of a leaf, shown with the mature fungus as a stroma, ascoma, and hyphal outgrowth anchoring the ant to the leaf.
The parasite we're about to learn about happens to be the most successful eukaryotic pathogen that affects about 30% of the world's population. Most people are unaware they even have it, as the infection causes no noticeable symptoms in healthy individuals. People with weakened immune systems may experience the following. So what is this parasite? Toxoplasma gondii is an obligate intracellular parasite that affects warm-blooded vertebrates such as mammals and birds. They grow and reproduce inside the cells of their hosts. T. gondii is a crescent-shaped parasite with the nucleus centrally located. Toxoplasma gondii belongs to the Apicomplexa phylum, which are a group of intracellular parasites with a complex life cycle. T. gondii reproduces mainly through clonal reproduction in the intermediate host, but its primary sexual reproduction occurs only in the intestines of cats, which is the parasite's definitive host. Other members of the Apicomplexa phylum are Plasmodium, which causes malaria, and another member is Cryptosporidium, which causes cryptosporidosis, an illness that causes diarrhea, which, similarly to T. gondii, is spread through fecal matter, contaminated food, and water. T. gondii is a chemoheterotrophic pathogen which uses organic compounds for energy and carbon content. T. gondii specifically relies on the sugar content of its host. By process of glycolysis, the parasite breaks down the glucose as a carbon source and for ATP synthesis. T. gondii's life cycle starts in cats, their definitive host. This is the infection stage where oocysts are shed in the feces of the infected cat. It takes 1 to 5 days for the oocysts to sporulate and become infectious in the soil, to the environment, and to other mammals and birds. The second stage occurs in the intermediate host, which is any host other than the cat. This infection occurs by accidental ingestion of contaminated food, so undercooked meat, water, or soil. Once inside the body, the oocysts hatch into tachyzoites. During the tachyzoite stage, the parasite is quickly multiplying. This infection stage is quickly spread through the blood and tissues, manipulating the immune system. This stage is usually harmless to humans with healthy immune systems. This parasite creates behavioral changes in mice by altering their neurotransmitter balance, impacting decision-making, and they lose their fear response, making them susceptible to easily being caught. And they can also become attracted to cat urine and have the tendency to explore, which exposes them to danger. The immune system eventually controls the tachyzoites and pushes them into a dormant stage where they become bradyzoites. During this stage, the bradyzoites form cysts in the muscles, brain, and organs and become latent. These cysts can stay in the body for life, causing no symptoms. However, if a human were to become immunocompromised, the infection becomes opportunistic and can cause harm. The cycle completes by being transmitted to a cat again, where the parasite undergoes sexual reproduction in the cat's intestines, creating new oocysts, which are then shed in the cat's feces. Some other transmission routes include pregnant women who have never been exposed to the parasite. It can be fatal for their newborn baby, their unborn baby, and that transmission is called congenital toxoplasmosis. You can also get it through organ transplant or blood transfusion. Toxoplasma gondii is considered metabolically flexible by being able to maintain its metabolism by adapting to different nutrient conditions. T. gondii will use the amino acid glutamine to produce pyruvate through glutaminolysis. It will also use glucose metabolism byproduct lactate and convert it into pyruvate as well when sugar sources are low. Interactions with other microorganisms will be the following. T. gondii has evolved to manipulate the human immune system by secreting proteins that interfere with the host cell signaling pathways. These pathways are regulated by small proteins called cytokines. Cytokines are responsible for cell functions such as growing, moving, or dividing. These tiny proteins have a big job of setting off the immune system's alarms to defend itself against foreign invaders. Due to the manipulation of T. gondii, the cytokines, the body then identifies the parasite as a non-threat, which allows the parasite to live undetected in its human host. Toxoplasma gondii is an interesting and complex protozoan parasite with a global presence. This book explores its biology, starting with its distinctive shape and anatomy, which help it infiltrate and reproduce in the host cells. We discuss the ties with the Plasmodium species, which cause malaria. Metabolically, T. gondii's ability to adapt to different nutrient sources is linked to its ability to manipulate the host cells, including influencing gut microbiota. As an adaptable parasite with intricate mechanisms, it's no wonder this parasite is as successful as it's been.
Plasmodium. Plasmodium is a parasite, a type of eukaryotic single organism of the genus Protozoa, which is transmitted by the bite of an infected female Anopheles mosquito, causing a serious life-threatening disease called malaria. Malaria is especially prevalent on tropical and subtropical areas. Plasmodium has an interesting complex structure and anatomy. It has an elongated shape, usually between 2 to 2.5 micrometers in length and about 1 micrometer in width. It is composed of two cellular membranes, an outer cell membrane called plasmalemma and an inner cytoplasmic membrane called glycocalyx. The plasmodium also possesses several unique organelles, including Golgi apparatus, endoplasmic reticulum, and mitochondria, and a specialized organ called apicoplast, which is essential for the synthesis of proteins and lipids needed for the parasite survival. Plasmodium belongs to the phylum Apicomplexa, which includes organisms such as Cryptosporidium, Toxoplasma, and Cyclospora. The closest relatives to Plasmodium are Cryptosporidium parvum and Toxoplasma gondii, both of which belong to the same genus and are closely related in terms of genetic makeup and life cycles. The Plasmodium itself is further subdivided into several species, including Plasmodium vivax, falciparum, malaria, and ovale. Each of these species is capable of causing different types of malaria in human host. Plasmodium require specific nutrients for growth and metabolism. Sources of nutrients include proteins, carbohydrates, lipids, vitamins, and minerals. The Plasmodium parasite obtain proteins from their host red blood cells and vitamin and minerals from the host plasmoserum. The metabolism and growth is highly dependable on the environment and the availability of nutrients. This parasite uses glucose as its main source of energy to generate ATP molecules that are used in the growth, replication, and metabolic processes. It's also capable of perform both aerobic and anaerobic respiration depending on the availability of oxygen. In terms of growth, Plasmodium is able to undergo asexual reproduction through a process known as schizogony, in which the parasite replicates a genetically identical daughter cell that is released into the host bloodstream. In addition, Plasmodium is also capable of undergoing sexual reproduction through a process known as gametogony. In this process, the parasite produces gametocytes that will eventually undergo meiosis to form haploid gametes that can fuse to form a zygote. The life cycle of the malaria parasite is complex. Plasmodium spends part of its life cycle in a human host and the other part in a mosquito. In the human host, the Plasmodium life cycle begins when an infected female Anopheles mosquito injects the parasite into a human's skin during a blood meal. The parasite then moves through the bloodstream, where it invades liver cells. This is the exoerythrocytic phase. Once inside the liver cells, the parasite multiplies to form thousands of daughter cells known as merocytes. Once the daughter cells are mature, they burst out of the liver and enter the bloodstream. This is where the erythrocytic phase starts. Here, the parasite invades the red blood cells, multiplies, and bursts out of the red blood cells, entering the bloodstream once again. At this point, malaria symptoms are evident. Some of the parasites differentiate into sexual gametocytes at this point. This is where the gametocyte phase starts. These gametocytes are taken up by another Anopheles mosquito during a blood meal, and the Plasmodium life cycle begins in the mosquito host. The parasite multiplies inside the mosquito midgut wall, and while in there, the microgametocytes fuse with macrogametocytes to form zygotes. The zygotes turn to oocytes that grow and rupture and release sporocytes, which make their way to the salivary glands of the mosquito to inoculate into the next human host. Plasmodium can also interact with environmental factors such as water, temperature, and humidity, which influence how it breathes, spreads, and affects health. Overall, Plasmodium and its interactions with other organisms and environmental factors are complex and dynamic. Its ability to avoid detection, persist, and spread within ecosystems. It's an important factor that has enabled its survival as one of the most dangerous human and animal parasites, affecting millions of people across the globe every year. In the year of 2021 alone, it was estimated that about 240 million cases of malaria happened. Most of these cases were presented in the African regions, affecting children under the age of 5. Plasmodium acts in a symbiotic relationship with the mosquito vector and the human host, benefiting from both. Pathologically, the Plasmodium has a detrimental effect on the health of the infected individual, causing symptoms such as fever, malaise, chills, vomit, anemia, jaundice, sometimes neurological complications, and even death, mainly also affecting infants and children under the age of 5. The environmental impact of Plasmodium is considerable, and it makes it difficult to mitigate. Populations of infected people and the associated mosquito vectors are usually confined to regions, leading to localized outbreaks. Additionally, due to the significant amount of resources that must be allocated to address the disease and its associated morbidity and mortality, governments in endemic regions are often forced into poverty. Also, due to the significant amount of insecticide used in order to reduce the population of Anopheles mosquitoes, runoff from application sites can be adversely affecting local ecosystems. In conclusion, Plasmodium malariae is a significant infectious disease with far-reaching implications for human populations, particularly those in tropical and subtropical areas. It can cause both symbiotic and pathological effects with possible dire health outcomes, and can also have environmental costs due to the resource expenditure, vector control, and vector killing runoff.
Hello. Today we're talking about Giardia intestinalis. We're going to start by going in the way back time machine to 1681, when Antony von Leeuwenhoek took a look at his own stool under a microscope. He saw tiny little creatures moving very prettily with sundry little paws making such a stir. He was looking at Giardia intestinalis. Zip zoom forward, and it wasn't until the late 20th century that Koch's postulates could actually be satisfied for giardiasis, the infection caused by this protozoan parasite. It's an external parasite, meaning it doesn't enter the body, but replicates in the digestive tract of its host. and each year about 280 million people worldwide have symptomatic infections. The life cycle of this parasite has two primary stages, the trophozoite stage and the cyst stage. Post-infection occurs when cysts are ingested. When the cysts reach the stomach and duodenum, they are exposed to pH changes and bile that trigger excistation, in which the ingested cysts differentiate into motile trophozoites and begin to replicate. Further down the digestive tract, some trophozoites transform into highly modified cysts in a process called encystation. Cysts get passed from the host feces to the environment and ingested by a new host, starting the cycle all over again. Now that we know about the two stages, let's talk anatomy. Trophozoites are pear-shaped with two symmetrical nuclei. They've got a ventral disc for adhering to host epithelial cells. They've got 4 pairs of symmetrical flagella for motility, and a median body, sometimes called the crooked smile, with an unknown function. Cysts are oval-shaped with 4 tetraploid nuclei surrounded by a wall. And here is the watercolor painting I made of Giardia intestinalis. Alrighty, on to phylogeny. Giardia intestinalis is in the order Diplomonadida, whose members tend to have 2 nuclei and bacterial-like characteristics, probably as a result of horizontal gene transfer. It's in the family Hexamedidae and the genus Giardia, which has 8 different sequence genotypes, but all are found in mammals and are host adapted. Okay, metabolism. Trophozoites have an anaerobic metabolism. They generate energy via glycolysis and arginine dihydrolase pathways. One of the reasons that cysts can last for weeks or months in the environment is that they only have 10 to 20% of the metabolism of trophozoites, so they conserve resources. Nutrition. Giardia intestinalis has a highly reduced genome, transcribing nearly 99% of its genome in lab studies. That is likely due to its parasitic lifestyle, which has allowed it to eliminate non-essential genes and simplify its organelles and cellular processes, relying on the host instead. So it lacks mitochondria and other canonical eukaryotic organelles. And it's highly dependent on nutrient uptake from its hosts via material exchange organelles called peripheral vacuoles. Populations grow via asexual binary fission. Moving on to interactions. It influences the microbiome of its host gut and vice versa. Giardia intestinalis inhibits nitric oxide production, which has an antimicrobial function, including against itself, so that helps with its own survival. It also alters the mucus layer in the gut, leading to proliferation of certain types of bacteria such as E. coli, and it decreases other types of bacteria. And some studies show that it can dislodge bacteria from biofilms in the gut, leading those dislodged bacteria to become pathobionts. Interestingly, studies show greater presence of interleukins and antibodies and adaptive immune response in mice with healthy microbiota versus germ-free mice. This may indicate that the presence of gut microbiota influence the host's immune response and clearance of Giardia intestinalis. Giardia intestinalis's effect on the environment is significant, including, as mentioned, mucus disruption, shortening of the brush border microvilli, apoptosis of epithelial cells, and compromised tight junctions. This slide is a really awesome summary of some of the things that we talked about, and there are a lot of things going on here. Finally, pathology. Infections are spread via the fecal-oral route, such as contact with cyst-containing feces or cyst-contaminated food and water. Diagnosis can be made by stool specimens or rapid fecal antigen detection tests. The infection can be asymptomatic, or it can include symptoms such as diarrhea, abdominal pain, fatigue, bloating, and weight loss. Acute infection usually lasts 2 to 3 weeks, and chronic infection has been linked to irritable bowel syndrome and chronic fatigue syndrome. Both innate and adaptive immunity work to clear Giardia intestinalis, and treatments are usually nitroimidazole, which are antibacterial and antiprotozoal drugs. That's it, thank you.